Spread of cancer cells from primary tumors to form the metastases at distant sites is responsible for the majority of deaths in cancer patients. Positive and negative regulators exist for metastasis, and understanding those regulators may lead to new modalities of cancer diagnosis and therapy. In 1996, researchers set out to determine the genes responsible for metastasis suppression. Genes were identified by searching for a 10-fold increase in their expression in non-metastatic compared with highly metastatic melanoma cells. One gene was expressed only in non-metastatic cells. Cells engineered to express different levels of this gene were tested for metastatic ability in mice. When different cells were injected into mice, those cells expressing relatively less of this gene resulted in more lung mets. And those expressing more of this gene had less lung mets. The gene named KISS for its role as a suppressor sequence of metastasis, with acknowledgment of the gene's discovery in Hershey, Pennsylvania, and the Hershey chocolate kiss. The irony of the name KISS and the role of the gene in reproduction would go unrecognized for 7 more years. Clinical evidence supporting the role of KISS in cancer has been seen in multiple tumor types. Patients with tumors having lower KISS have a higher probability of disease recurrence and shorter overall survival. For example, in gastric carcinoma, patients' KISS was the strongest prognostic marker of overall and disease-free survival. KISS is also found in some normal human tissue. In women, the placenta expresses so much KISS that it spills out into the blood where it can be detected throughout pregnancy. KISS may regulate invasion of the placenta and the uterus, much like it regulates the spread of tumors. Understanding this may help us understand cancer better. KISS and its receptor are also in the brain. More importantly, they're in the hypothalamus, the seat of the neuroendocrine system, where the brain is affected by and affects reproduction, growth, metabolism, and other endocrine-related functions, strategically and ideally located to affect important physiological processes. The central function of KISS in regulating reproduction went unnoticed until 2003, when scientists reported the presence of a mutation in the KISS receptor in consanguineous, or inbred, as we say in Kentucky, humans suffering from syndromes characterized by absent pubertal development. They didn't go through puberty, and I'm from Kentucky, so I give myself some liberty. Mammals with inactivation of the KISS receptor are sterile and have grossly underdeveloped reproductive systems, very small testes and ovaries. Whether the effect on reproduction was occurring at the brain and the hypothalamus, the pituitary, or the ovaries or testes was unclear. Now we have to review the hypothalamic pituitary gonadal axis and how it regulates reproduction. From the brain, the hypothalamus secretes gonadotropin-releasing hormone, or GnRH, into the local blood supply. That in turn stimulates the release of the hormones from the pituitary gland, and these affect the release of sex steroids and gametes from the testes and ovaries, eggs and sperm. Research involving both inactivation of the KISS receptor and administration of kisspeptin, the protein product of that gene, have proven that KISS neurons in the hypothalamus control the release of GnRH, making KISS the supreme regulator of reproduction. Without it, none of you would be here. There has not been a more important discovery in the area of physiology and medicine and science in this area of reproduction in more than 30 years. Since many of these releasing hormones from the hypothalamus, like GnRH, were discovered by researchers like Dr. Shally and Guillemot, who earned the Nobel Prize. In effect, this has opened a black box. For more than 30 years, the hypothalamus has been that black box, acting as a stoplight or governor of reproduction, while inputs like sex steroids from the gonads, energy from food eaten, and photoperiod related to season clearly affect reproduction. The mechanisms have remained a mystery until now. Estrogen from the ovaries obviously have positive feedback within the hypothalamus, leading to ovulation. These KISS cells of the hypothalamus appear to be the missing link that receive and transmit sex steroid feedback to the hypothalamus, because KISS cells express these necessary steroids so they can detect progesterone and estrogen. There's compelling evidence that KISS provides a trigger for the onset of puberty. KISS cells act like a brake before puberty and an accelerator after puberty in response to these steroids. Furthermore, some cases of precocious or early puberty have been associated with aberrant KISS. KISS expression in the brain also changes with reproductive cycle. This is particularly important when we study domestic species like cattle. For instance, dairy cows are thought to be the goddess of infertility, and if we can understand KISS expression in these animals, we could help reproductive performance in these animals and our farmers. Even before 25,000 BC, when the woman of Willendorf was thought to have been carved, we knew reproduction was a luxury, not to be attempted with inadequate nutrient availability. We now know that the KISS neurons are affected by leptin, the hormone made by fat cells, which integrates the signaling of body energy reserves to allow the animal to go to reproduce or to hold it back. We have also shown that KISS is a coordinator of energy balance and metabolism in domestic livestock. Study of KISS during lactation in dairy cows and through specialized brain catheters we've placed in sheep has yielded critical insight into the mechanisms regulating reproduction in the growth axis and how they work together. KISS may be a novel therapy for infertility. KISS is a powerful stimulus of reproduction. In the face of food restriction in the mice shown here and seasonal infertility, which is normal in sheep, when these animals don't reproduce, if they're given KISS, they can drive the animals to proceed through puberty and have offspring and ignore the normal signals. To the contrary, KISS may also become a target for contraception. By eliminating the KISS gene or the KISS neurons, reproduction may be stopped. Ongoing work in our lab to silence the expression of the KISS receptor through siRNA in the brain cells may lead to new and novel ways to control reproduction. Thank you.